Today we're going to talk about chromosomal mutations and how they occur. We will identify and illustrate changes in DNA and evaluate the significance of these changes. So what we're going to do is find changes, locate them, see what they look like in DNA, and then we're going to, through models at the end of class, we're going to illustrate those changes so that you know exactly what they look like. You've got copies of that in your notes. We're going to create a model at the end using letters and number patterns so that it's a little easier to recognize, so those mutations are easier to recognize. All right, here we go. So for chromosomal mutations, it's a change in DNA. A mutation can happen at the nitrogen base level, so you could substitute out an A for a G, or it can happen at the chromosomal level, which is what we're talking about today. So it's a little bit larger sections of DNA that we're talking about mutating today. A chromosome is condensed DNA. This X shape is what you usually see inside the nucleus of a cell. If you look up here, you can see those little tiny X shapes up there. That's condensed DNA that's ready for cell division. So it's nice and compact and organized and ready for the cell to move it around in cell division. Changes to the chromosome can happen during cell division, causing the DNA to get rearranged, and this is a form of mutation. So if you take a piece off, that's a certain type of mutation. If you insert a piece and shift everything down, that's a kind of mutation. If you flip-flop a piece around, like take it off and turn it and stick it back on, that's a type of mutation also. And all of those have effects on the traits of an organism. Okay, chromosomal mutations occur when big pieces of the chromosome are altered or changed in some way. These mutations usually occur during meiosis, when eggs and sperm are being produced. So remember, meiosis, that is cell division to produce gametes. Remember, these are called gametes. And gametes are just sex cells. There's 4 types of chromosomal mutations. Deletions, which is exactly what it sounds like. If you hit the delete key on your computer, that takes a piece out, takes a word out. Inversions. An inversion is just when you switch something around, turn it around. So you're going to invert it. Excuse me. Duplications is where you take a section and you duplicate it. So you're going to make another piece that looks just like it and insert it into the chromosome. And translocation is where you take a piece off of one chromosome, you take a piece off of another chromosome, and you switch them. And you'll see examples of those here in a minute in the notes. So here's the deletion that we were talking about on the previous slide. When part of a chromosome is removed, you can see this letter D here, A, B, C, D, E, F, G, H. We're going to take that D out. So we're going to delete the D and then put all of the chromosome back together. So you end up with A, B, C, no D, E, F, G, H. That deletion has happened right here. The next type is an insertion or duplication, when part of a chromosome is added or copied. So we've got our BC that we're focusing on for this chromosome. We're going to take that BC and copy it, so that when we look at our new chromosome after the mutation has happened, we have 2 BC sections, A, B, C, B, C, D, E, F, G, H. And you can see that that is not the same as the original chromosome. Okay, an inversion mutation is when parts of a chromosome switch order or place. So we've got B, C, D here we're going to work with on this chromosome. We're going to take it out and then turn it over and then put it back in so that it's in the opposite order. It's in the reverse order from when we started. So if we look at our complete chromosome, we have A, D, C, B, so this part is backwards, E, F, G, H. Remember, each of these letters, they are basically genes, segments of your DNA that are going to code for a trait. So if you flip-flop those around and you go through protein synthesis with this piece of DNA, you make mRNA and it goes out to the ribosome. That's going to create a different protein than the original strand would. Translocation is the last example, when part of one chromosome breaks off and is added to a different chromosome. So you can see here we've got our crisscross chromosomes down here at the bottom, and they're going to break off and reattach to opposite chromosomes. So instead of having A, B, C, D, E, F, now we have A, B, C, J, K, L. And then we have, instead of G, H, I, J, K, L, we've got G, H, I, D, E, F. So those pieces have flip-flopped, and that's definitely going to end up with different proteins. Okay, quick question. Think about the 4 ways chromosomes can be mutated. Which type of mutation do you think is the most harmful? Why do you think that's the most harmful? So take a second here, talk it over with your neighbor. Go through and think about these. Which one do you think could be the most harmful? Okay, so I've given you about 30 seconds to talk it over with your neighbor. We're going to go ahead and move on. Another way that chromosomal abnormalities can happen is through non-disjunction. We talked about disjunction when we were talking about meiosis. If you remember, disjunction was when the chromosomes separated in anaphase, so non-disjunction would occur when they stuck together and they didn't split properly during anaphase. Okay, so here's the definition, non-disjunction. Homologous chromosomes or chromatids do not separate equally during meiosis. So you see the splitting doesn't occur properly. Gametes will have too few or too many chromosomes because when that one splits and goes to one egg cell and it takes it away from the other egg cell, that's going to present a problem for those egg cells. The resulting offspring will have an abnormal chromosome number because if a egg cell that has one extra chromosome in it gets fertilized by a sperm cell with a regular number, you're going to see that the number in the end would be different. So if our egg cell had 23 and our sperm cell had 24, so that's an extra one, then we'd end up with 47 in our offspring. And you can see over here, this is called a karyotype over here. It's just a picture of all the chromosomes in an individual. Most karyotypes have 2 chromosomes for each number, so you can see here like 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, and so on and so forth. When you look on down, you can see here there's 3 chromosome 21s, and that is not a normal situation. Typically, you have 2, and so when you have this extra one, it results in different characteristics. And 3 chromosome 21 sometimes is called trisomy 21. That tri means 3, and the everyday word for trisomy 21 is Down syndrome. So if you have 3 chromosome number 21s, then you end up with a Down syndrome offspring. Okay, here's just a pictorial representation of non-disjunction. You can see here this is metaphase 1 of meiosis, and the chromosomes are lined up across the middle of the cell. Remember, metaphase and middle, M and M. When they're pulled apart, you can see here these 2 big guys went to the same side of the cell here in anaphase. So that's how you end up with extra chromosomes like you see over here on the end. So this is N plus 1, so like 23 is our N number plus 1, so this one would have 24. N plus 1 again, 24. N minus 1 would be 22, and N minus 1 would be 22. And that obviously is just in humans. Non-disjunction occurs during anaphase 1 or anaphase 2 of meiosis. The homologous chromosomes fail to separate equally, so you can see here we've got 1 on this side and 3, where typically we would have 2 and 2. Okay, quick question. Describe the significance of anaphase to meiosis. So remembering, I'm going to flip back. So remember, anaphase is here, and that's when the chromosomes are pulled apart to the sides of the cell. The significance of anaphase to meiosis, and what could occur if anaphase is not completed properly. So take a minute, think on your own, talk to your neighbor, come up with an answer. Okay, so you've had a little bit of time to think. Remember, anaphase is when the chromosomes split and go to the poles of the cell. So you've got your little chromosomes here. This is our cell. And they're moving towards the sides of the cell. These guys are going to move out. And what could occur if anaphase is not completed properly? Well, that is non-disjunction. And because of non-disjunction, we can end up with Down syndrome. Trisomy 21, you can end up with lots of different chromosomal abnormalities. Okay, here's a YouTube video we're going to watch real quick.